Hi everyone, welcome to Blood Bank Lab 4. So with this lab, we're going to focus specifically on RH phenotyping. And so we did talk about other RH antigens when we did our review of the RH blood group system in lecture. So this should be some information that you probably have heard before. So remember, that big D is the antigen of the RH system that confers whether someone is RH positive or negative. And in our last lab, we focused on weak D testing to detect weakened expression of the D antigen. And so today we're going to be looking at other important antigens of the RH system. So with regard to transfusion, these other antigens include big C, big E, and little e. Most individuals have at least one type of C, meaning that you inherit at least big C or little c. It would be highly unusual not to have either one of these. And most people also inherit at least one type of E. So either big E antigen or little e antigen. And once again, it would be highly unusual, very rare to lack both big E and little e. Okay? So we did learn in our lecture Zoom that the absence of all RH antigens is referred to as RH null. And so that word null means zero. And this absolutely does have an impact on the red blood cell membrane. It leads to instability of the membrane and shortened lifespan of the red cells because the membrane basically is unstable and leads to early cell death. So these RH antigens really do serve a purpose. Phenotyping is the term we use for testing when we're trying to determine which antigens are present on red blood cells. And to do phenotyping, we use known antisera directed against each specific antigen. So this is really very similar to forward typing that we do with ABO. Okay, so just like we have anti-A that detects the A antigen, anti-B that detects the B antigen, we have other reagent antisera, namely anti-C, which is antibody against big C antigen, anti-little c, anti-big E, and anti-little e. And so these are the reagents, the knowns, that we use to determine if the corresponding antigen is present on a person's red blood cells. So similar to other tube testing interpretation, when we have agglutination in a tube, it does indicate that the corresponding antigen is present. So for instance, if I had agglutination in the tube with anti-little c, then that means that my patient has the little c antigen. If it lacks agglutination, then it means that that antigen is absent. So for our RH antigen typing procedure, it's really very simple. We would label 4 tubes, one for each of our antisera that we're going to test. So we have one for C, for little c, for big E, and little e. And then what we're going to do is add 1 drop of the corresponding antiserum into the tube. Okay, so 1 drop of anti-C here, 1 drop of little c, 1 drop of anti-E, and 1 drop of anti-little e. Next, we're going to add the very same 2 to 5% red cell suspension that we prepared on our patient for forward and reverse ABO typing. It's the same solution, so that's nice. We don't have to make anything new. So we're going to add 1 drop of our patient's 2 to 5% red cells into each one of these 4 tubes. And we're going to give these tubes a brief mix and place them in our centrifuge. This is what we call immediate spin testing, IS, right? So immediate spin, we add everything and we centrifuge immediately. So we're going to centrifuge for 15 seconds at 3,500 RPMs, and then we're going to remove our tubes, take them out, put them back in order in our rack, and we're going to read them. So this is a picture of what the antisera look like, and so you can see, you know, they're just bottles and they have the labels on them. So it's important that you read the labels for the different antisera. Make sure you put the appropriate one in its corresponding tube. So when we're doing our reading, the same rules apply. Okay, we're going to have our buttons down in the mirror. We're going to use our viewing lamp. We want to be very gentle when we're mixing these because they are not going to be 3+ or 4+ most often. They're going to be weaker reactions. So you don't want to mix them, you don't want to miss them. You don't want to overmix them. So as you're reading, you want to grade any positive reactions. If there's no clumps, keep mixing and reading until the button is completely gone, and record as negative. And once again, as mentioned on a previous slide, if both the big C and the little c are negative, or both the big E and the little e are negative, this would be extremely rare. So what we would do in an instance such as this, we can take the tubes that are negative and just recentrifuge them again, put them in the centrifuge for 15 seconds and read them again, and be careful not to overshake our tubes. And oftentimes then we might get, like maybe a 1+ reaction that we missed the first time. Okay, so you have to have at least one kind of C and you have to have at least one kind of E. It would be very, very unusual not to have that. So you guys know all about grading reactions, and so on the previous slide I mentioned to you guys, it would be unusual to get 4+ or even 3+ when we're doing the phenotyping. So most often it's going to be 2+ or 1+ reactions for your positives, so you have to be very careful that you're not overmixing them when you're reading. So on the next slide here, I prepared an RH phenotyping chart of maybe how we would record results for testing. And so what we see here is I have a column here for anti-D, and the results here with anti-D, remember, these are typically done as part of forward ABO and RH testing. We probably have these from previous testing, so we just want to carry the results down from our forward typing, okay, for the anti-D. And then we have our other 4 antigen tests here. Okay, so what I'd like to do, excuse me, I made up some examples that I would like to show you because you'll be doing this as part of your lab report today, of how to determine the most likely genotype by looking at a chart, knowing the phenotype. So the testing results that we have here is the phenotype. And then looking at known data on a chart, we can predict the most likely genotype, and then we're going to convert it into Wiener classification for practice. So the first example here, patient has 4+ with anti-D that we carried from forward typing, and then we had a 1+ reaction with anti-C, excuse me, big C, which means they have the big C antigen. There was no reaction with little c. There was a 1+ reaction with the anti-E, and also a 1+ reaction with the anti-little e. And so these blood system antigens, or these RH blood group antigens, are inherited similarly to ABO. So it's entirely possible, to have 2 types, both big C and little c, or both big E and little e, as we see in this example, because they exhibit codominance. Okay, so it's entirely possible to have more than one. So what are we going to do next? Okay, we're going to move ahead to our next slide. And I have a copy. Let me just go back 1 minute. So if I look at this testing here, we see the only antigen this person lacks is little c, and that's going to be helpful when we're looking at our data. So this is known data chart that I copied over from a procedure that I'll show you, is also loaded under Canvas if you'd like to look at it. And so basically on the left-hand side here, we have different phenotypic results from testing. And so when you see a positive, it means the antigen was detected, and when you see a 0, it means it was not detected. Okay, and so in our example, let me double check, the only one this first person does not have is little c. So I basically, want to go down through the rows. Like this one wouldn't fit, they don't have big E. So this one, they don't have little c, but they also don't have big E, so that would not fit our test results. We see in the next line, it's entirely possible to have all of them. We get down, we're getting closer. Okay, here, this one, they're lacking the big C. Okay, and so we have to keep looking until we find the one where they're only missing little c. So these wouldn't work because they don't have big D. These would be all RH negative people. And finally, when I get down here to this one, let me circle it. Let me do my annotation. Okay. See this one? So if we look at this one, let's see if it checks out. Okay. So big D, big C positive, big E positive, no little c, and they have little e. So this fits the exact same pattern that we got in our testing of our patient. And so what we're going to do then next is we're going to go to the column to the right, and this lists possible genotypes for a patient with this phenotype, okay? And the one that you see listed first, okay, you see how this one is listed first? That is the most common one. So this is the one that we're going to copy over to our chart, okay? So we're going to get out of here. Okay, so it's big D, big C, big E, and then big D, big C, little e. Okay, so let's go back. Okay, so let me just clear my drawings. Okay. Okay, so I'm just going to type it. Oops, my mouse is acting funny. I can't, that's why. Okay, so let me get out of slideshow and we'll type it. Okay, so based on the chart that we looked at, okay, what I'm going to write here is the first genotype. Okay, so it was big D, big C, big E slash. So remember, this first one is inherited from one parent, and then the second one is inherited from the second parent, and that was big D, big C, however, with little e. Okay, so we're going to convert this to Wiener classification. So hopefully you remember these rules from how we practice these in lecture. So remember, whenever you have the big D, I'm going to write a capital R. Okay, so there's definitely a capital R here, and for this first example, there is both big C and big E. So I'm actually going to write, it's a Z for both of those, right? That's kind of unusual, and this would actually be a superscript if I could figure out how to do that. Let me see if I can do it. Yeah, like that, okay? And then we're going to look at our, from the second parent, it's also going to be a big R, okay, because there's a big D. However, here, the only capital letter is big C. So remember, when there's only big C and no big E, it's called R1. So we would write, superscript again. Okay, so this one would be RZ R1. Not a very common phenotype, but hey, we need practice so you guys can practice with the classification of Wiener. Okay, so let's look at our second example. When we look at the second example, we can see that this person is Rh negative from our forward typing testing for D. Okay, so they lack D, they lack big C, they have little c, okay, 1 plus, they have big E, and they have little e. So they have 3 antigens here. Okay, so just like last time, we're going to go to our chart. And maybe I'll make this a little bigger. I'll go into slideshow for a moment. Just so you guys can see it. Okay, so remember, this first whole bunch we're going to skip because we have an Rh negative person that lacks big D. So we look here, okay, this one, okay, we also need to have the big E, which we don't. Not this one, not this one. Let's see. Nope. Okay, let's see. Big C, big E. Nope. Let me just go back and check. So they have only little c, but both e's. Okay. Oops, wrong way. Sorry about that. Okay, so we're looking for D negative with only little c and both e's. Okay. I might have skipped it. Okay, let's see. Ah, yeah, no, that one doesn't fit. Okay. Aha, I skipped over it. So it looks like it's going to be this one. Let me get my pen. So if we look at this one, okay, they don't have. So they lack big D, they lack big C, they have big E, they have little c, and they have little e. Okay, so that should fit the results of our testing. And for this one, we're lucky. There's really only one possible genotype that's listed on our table. So we're going to copy this down on our we're going to go back to our chart. Okay, so I'm going to clear my drawings. Okay, and I'm going to get out of here and get out of slideshow. Okay, and then we'll go back so we can type in our results. Okay, so no big C, no big D, but little c, big E, and little e are all there. And so remember, sometimes people will write little d to indicate the absence of big D, but there really is no little d antigen. Okay, so little d, little c, big E. And then the second choice was little d, little c, little e. Okay, so let me just go back and check to make sure. Okay, yeah, little d, little c, big E, and little d, little c, little e. Okay, I think I'm right. Yes, okay. So how are we going to convert this to Wiener? Remember, little d means little r. Okay, when we look at this first one, there is only big E. And when we have big E, it is double prime or a number 2. Okay, so here we're going to write double prime, okay, because it's the lowercase r. And then the second one, we're going to write little r again, but there are no other capital letters, so it's just going to be this, r double prime, little r. Okay, so we're going to just finish up this presentation. Just wanted to point out to you all that the RH phenotyping can also be done using the gel card method. And so basically, Ortho makes gel cards that, let me blow this up, makes gel cards that have the anti-sera already placed into the individual cups. So you see them labeled here. So this one would have the anti-big C, the anti-little c, anti-D, anti-big E, and anti-little e, and this is a patient control that's on the card as well. And so it really just entails adding the appropriate dilution of the patient red cell suspension to each one of the cups. And the card is going to be centrifuged at immediate spin, meaning just like with tube method, you add everything and you centrifuge. However, with the gel method, when we finally get a chance to do a couple practice labs with this, the gel card centrifuge for 10 minutes. So it's a much longer centrifugation process. So I'd just like to review reactions in gel with you guys because it's been a while since we've talked about them. So remember, with gel, when an immune complex forms, so in this tube here we have anti-C that found its target big C antigen on the patient, and we see the 4+ reaction lays on the top of the tube. So it's the opposite of you know, the regular classic tube method of testing. So this would be a 4+, meaning that this patient has big C antigen. A negative, everything's going to sink to the bottom. So there's no little c antigen. So there's a 4+, they're Rh positive, they have big D. There is no big E, and then a 4+ with little e. So we can see that this patient does have at least one type of E, they have little e, and one type of C, they have big C, along with the big D. And so they lack little c and big E. So let's go back and find their genotype. Oh, geez, you know, I should write this down. They lack little c and big E. Brain is fried, guys. Okay, so it's going to be D positive. They don't have big E, but they also don't have little c. Aha! What do you guys think? Big D, big C, and little E? Could it be? Ha ha ha. Let me circle it. This one, right? Let's just go back and make sure that I did this correctly. No big E and no little c. No big E and no little c. Okay, so that's basically the phenotype, and we just happen to get the phenotype using the gel method instead. And so in this case, the most likely genotype would be this first one, and it happens to be big D, big C, little e from one parent, and the same exact haplotype, big D, big C, little e from the second parent. And so if you were with me, I would be quizzing you. Big D, let me get my pen. This is going to be R. Has big C, it's going to be R1. And it's the same thing here, big R1. Sorry, hard to write with this pen. So basically that would be the most likely genotype, okay, with our gel card method. Okay. So I am just going to clear my drawings, okay? And I think that's the end of our PowerPoint. It is, but I am going to show you guys some other things. So I'm going to get out of the PowerPoint and stop the share, but I'm going to show you some other things that are going to be important for lab today. Okay, bear with me, guys. I'm sorry. You guys know from previously that unfortunately for me, what I've clicked on most recently is what shows up to share. So is it here? Not really, but yes, that's what I want to show you. Okay. So when, you know, hopefully you have gone into Blood Bank Lab 4 already. So the PowerPoint's here, the recording link will be here updated for you with the code. And there are some supplemental lab videos to watch today. So there's basically one that is going to show you how to do the RH phenotyping, and unfortunately the only one that was on YouTube to look at was done by the slide method, okay? And so when you do look at this video, you're going to see that they're using the same anti-sera we talked about. However, they're doing the testing on a slide, okay, and looking for the agglutination that way. So it's a little bit different than how most people do it, but that was the only video that I was able to find, okay? You may want to open up this Lab 4 RH phenotyping procedure, which is a Word document. So I am going to show you what that looks like already opened on my computer. Okay, so when you open up this document, this is basically a written procedure that describes a little bit about these other RH antigens, some of which was in the PowerPoint. And this would be the procedure that we would be using for the tube method, which is similar to what I put in the PowerPoint as well. But it's nice to have this if we ever get a chance to do some labs together. And then this is the same chart, okay? If you would like to open this up to help you, you know, you're going to need this for the lab report, you know, as we did some examples, you know, finding our phenotypic testing and the most likely genotypes. You could either look at it here or from the PowerPoint. Okay, so that is available. And sorry guys, sorry. And so when you actually do go to do the lab, there are some other things you're going to be doing just, you know, besides RH phenotyping. So just like last time, you want to click on the link here. And so just like last time, you're going to be doing some blood typing with the blood typing game to maintain your skills of interpretation. And so I have to allow flash on my computer. You might have to do that too. And so I hate the sound, so I always block it. You know the deal by now. Proceed. We're going to be doing random patients again. And so basically, you know, you're going to be typing the 3 patients. And just like in lab 3, as you go through and you find out the types, you're going to be filling in your lab worksheet. Okay, so let me just stop here and go to the lab worksheet for a moment. Okay, so you want to download this lab 4 worksheet, which is also in the module, and I'll show you where. So here's the directions. Go to the blood bank game or click on the link like we did. And this is just as you did in lab 3. So you're going to test your 3 random patients. You're going to put your results in here, type them in as you are going and doing the game and the units you select for transfusion. So this is very similar to, it's exactly what we did in the last lab, okay? So other things you're going to do on the lab report, I have just made up some random results on 4 patients, and so you want to practice interpreting ABO. Here's our forward and reverse like last time, and there is RHD and RH control here, and some of these you can see have weak D testing results, so hopefully you remember how to interpret the weak D from last time. Remember, weak D is usually done when we have an RH negative person who tests that way with forward typing initially, and then we're going to carry the D tube and the RH control tube through the weak D interpretation. So you want to tell me the ABO and RH interpretation here for these 4 patients. Then when you get to the third part of the lab, this activity is just what we did together on the PowerPoint. And so basically I have typed up for 4 different patients some phenotyping results, and I did an example here. Okay, this is actually the one that was the gel card we did. Okay, I didn't know that. So you're basically going to have to access the chart either from the PowerPoint or from the procedure, the Word document, the known chart, and you're going to pick the first most likely genotype, type it in here, and then you're going to convert it to Wiener. So I did an example here. Okay, so you're going to do that for all 4 of them. And you guys know, always when you're done, save your work, okay? And then bear with. It's really stormy outside right now. Let's see if it'll, okay. So when you go back to your lab 4 module here, you know, you can see you would have downloaded your report here by clicking on the link, type it as you go when you play the game, save all your answers, and then when you're ready to upload, click on the assignment, and the directions are here. So basically yours would look a little bit different. You're going to, you know, follow the directions and upload your completed report, and it's worth 25 points as usual. So good luck, and be safe, and I will talk to you next time. Take care.